Can somebody please confirm you can hear me?
We can hear you.
Thank you. I think we're on chapter 4. Let me check on that. Yep. Okay, before we begin, are there any questions? Uh, hmm, sure what's going on here. All right, you should be seeing my screen now. Today is October 21st, and we should be finishing chapter 4 today. We have a quiz on Tuesday, quiz 2. It'll be covering labs 3 and 4, chapter 3, and the end of chapter 2. Any questions about any of that? I talked about some grading in the lab, or I will in the next lab.
Sorry, can you repeat what's on the quiz again?
It's on the syllabus. It's chapters 3 and the end of chapter 2, and then labs 3 and 4. All right, where were we? Oh, I like it when it opens to the last place you were at, but it's not doing that now. All right, we talked about damaging the prokaryote cell wall. Any questions about anything about the cell wall? If not, let's move on to the cell membrane. The cell membrane is a phospholipid bilayer. The main molecule is phospholipid. See if I can blow this up. Which is composed mostly of phospholipid molecules, shown here, the head and the tails. And they're a bilayer where the fatty acid portion of one is against the fatty acid portion of another, because these portions of phospholipid do not like to be around water or water soluble molecules. And so they come together. And then the heads face inward because they like to be around water, and the cytoplasm is mostly water. And the outside of the cell is also mostly water. Think of this as a blood cell. The blood is mostly water. Doesn't matter where you are in the human body, it is mostly water. So the outside of the cell membrane is also a head. There are proteins which are in the membrane. In fact, there's carbohydrates and lipids as well. But we're going to talk about the proteins. There are three types of proteins: peripheral proteins, integral proteins, and transmembrane proteins. A peripheral protein is a protein which is mostly either outside the cell membrane, or inside the cell membrane. Let me get that up there. So that's why it's called the peripheral protein, because most of the protein is on one side of the membrane or the other. The integral proteins and the transmembrane proteins span the membrane, like this pore protein, probably a channel pore protein, and that protein there, a transport protein. All of these proteins span the membrane. Any question about any of that? Now, for the membrane, let's talk about, for the protein in the membrane, the proteins which are mostly around water, like this peripheral protein, the amino acids in this portion of the protein will be ones which are mostly polar. And then the amino acids in the protein here, at least the R groups, are going to be nonpolar. And that's because the protein will have the nonpolar regions around nonpolar molecules like the fatty acids of the lipid bilayer, and then the polar parts of the protein around the liquids like the cytoplasm. And for a protein which is, I guess, not in the membrane but someplace else, it will fold itself so that if it's in water, the polar part of the amino acids or protein will be facing the water, and then the non-polar parts of the protein or amino acids will be tucked inside the protein, away from the water. And then the same with a protein in a lipid or let's say a lipid bilayer. These proteins here will have, at least on the outside, the non-polar amino acids or R groups, and tucked on the inside will be the polar R groups or amino acids. Of course, the polar amino acids can also be on the outside or inside of the protein too. Any question about any of that? All right. The plasma membrane follows the fluid mosaic model for our understanding. And the fluid mosaic model states that the membrane is a mosaic of phospholipids and proteins, and the membrane is as fluid as olive oil. So if you think of olive oil at room temperature, it's a liquid, a little viscous compared to water, but a little, well, definitely a liquid. You put olive oil in the refrigerator, it'll become mostly solid. And you put it in the frying pan, you heat it up, it'll become vapor. The point is that the cell membrane is a lipid, at least at our body temperature. And it's amazing when I think of it and look at my hand here. Humans only have, or animals only have, the cell membrane holding the cells together. And when I think of my hand, it's being held together mainly by a fluid, the cell membrane. Any question about any of that? That always amazes me when you think about it. Now, it's true the skin has an outer layer, which would be dry and mostly keratin. But like I said, the cell membrane is what's holding my hand together, and it is a fluid. The proteins can move about in the, or at least many of them, can move about in the cell membrane to function. Those of you who've had AMP, you know that the, oh, what are they called? The receptors will move together once they bind to their ligand, and then will come in as a group to send the signal into the cell. And that tells you that these proteins are moving about in the cell membrane. There's many proteins that move about in the cell membrane. The only proteins that do not move about are those that are anchored either to the intracellular matrix or to the extracellular matrix. So if this was a bone cell, for example, or bone osteoblast or something like that in the bone, this protein here could be anchored to the extracellular matrix, which would be the bone, in which case this protein will not move about in the cell membrane. And the same with this protein here, although it's anchored to the intracellular matrix, which we'll talk about that in a little bit, then this protein would not move about. But other proteins, like this one here, is free to move about the cell membrane. The phospholipids can also move laterally from one position to another, as well as rotate in position here. In fact, water actually moves into the cell membrane because water molecule comes up to the cell membrane. It cannot get through the phospholipid bilayer. But every once in a while, this water molecule will move over here, and then the water can slip in right there. It can't get through this layer of the bilayer, but this water molecule just stays there, and it occupies the space because the phospholipid won't want to be around where water is. And that water molecule just waits. Come on, mouse. just waits for this phospholipid to move over, and then the water molecule can move into the cell. Any question about any of that? Now, the entrance of water into a cell by this means is much too slow for the roots or most cells. So most cells have aquaporins, which are molecules. I think they're transport proteins, but I'm not sure they're a protein. They're an aquaporin, which pump water from one side of the cell to another. And in the roots, that water will be pumped from outside the cell to inside the cell. Any question about that? As far as I know, most cells have aquaporins on them because you do need water to move around in a cell, even in a plant cell. The roots move it inside the cell, but once the root has the water, it has to move throughout the plant, and how it moves into cell is by aquaporins. At least moves into cells quickly. Water moving across the cell membrane by waiting for a phospholipid molecule to move, and then the water moves halfway, and then it waits for the other phospholipid to move, and the water moves in. The water moving into the cell would happen too slowly. Any question about any of that? Making sure I covered everything. All right, the plasma membrane has selective permeability. This means the plasma membrane allows some molecules to pass, but not others. No large molecules can move across the cell membrane without the help of a transport protein. Ions either do not move across the plasma membrane or they move very, very slowly across the plasma membrane. Smaller molecules like water, oxygen, and carbon dioxide can move across the plasma membrane. Oxygen and carbon dioxide are the two most important. Oxygen can diffuse into the cell because oxygen is usually less inside the cell than outside the cell. And then carbon dioxide diffuses out of the cell across the cell membrane because carbon dioxide is higher in concentration inside the cell than outside the cell. Lipid-soluble molecules can move right across the plasma membrane because the bilayer of the membrane, meaning the fatty acid bilayer, is lipid. But if you think about it, there are not a whole lot of lipids in the blood. There are some, I'm not saying there aren't any, but most of the blood is water and water-soluble molecules. But those lipids which are in the blood can move into the cell directly, right through the cell membrane. Large molecules like proteins, DNA, even nucleotides and amino acid cannot move across the cell membrane without the help of a transport protein. Any question about any of that? All right, the plasma membrane contains some miscellaneous ingredients. For prokaryotes, this is where the majority of the enzymes are for making ATP. For photosynthetic prokaryotes, this is where the pigments are for engaging in photosynthesis. For the photosynthesis, the pigments are actually found in foldings of the cell membrane. So if we look at the cell membrane here, there's the cell wall. The cell membrane isn't flat across the cell wall. The cell membrane actually goes like this and then folds out, coming back, coming over, folding out, coming over, folding out. And in these roundish outfoldings, that's where we find the pigments for the photosynthesis, which if this is a microscopic algae, that would be chlorophyll. For what it is in bacteria, that's not chlorophyll. I'm not sure. Depends on the color for one thing. These foldings on the cell membrane, which is part of the cell membrane, remember that, are actually called chromatophores or thylakoids. If you damage the plasma membrane, you'll have leakage of the cell contents, meaning the cytoplasm will leak out of the cell, and that will kill the cell. You can damage the plasma membrane with alcohol. You can damage the plasma membrane with quaternary ammonium compounds and polymyxin B antibiotics. Polymyxin B puts holes in the cell membrane, and then the cytoplasm can leak out of the holes in the cell membrane. Any question about any of this? All right. The movement of materials across the membranes, like the plasma membrane in a prokaryote cell. But remember, it can be an organelle membrane in a eukaryote cell. Movement of materials across the membrane is critical to the cell's survival. The cell has two processes used in moving materials across the plasma membrane or across anything, actually. You can have a passive movement where the molecule moves down its concentration gradient. If the molecule is higher outside the cell, it will move inside the cell down its concentration gradient. And there's three processes. We'll talk about that in a minute. Passive processes. But there's an active process, and that's where the molecule moves against its concentration gradient. For example, if glucose is outside the cell, even if there's fewer outside the cell than inside the cell will want that glucose moved inside the cell. And the cell will move the glucose into the cell by an active process where the molecule will be moved against its concentration gradient from areas of less concentration to areas of higher concentration. Active processes require a transport protein on the cell membrane to literally pump that molecule of glucose into the cell. And for each molecule moved into the cell by an active transport protein, the cell has to use up one ATP molecule, which will be broken down into phosphate and DTP, diadenine triphosphate. No, excuse me, ADP, adenine diphosphate. And what the ATP is doing is giving the energy the transport protein needs to pump the molecule against its concentration gradient. Any question about the active process of moving a molecule into the cell? All right, let's return to the passive movement. So the passive movement, where the molecule moves down its concentration gradient, occurs by three means. Simple diffusion, where a solute moves from areas of higher concentration to areas of less concentration. Facilitated diffusion, where once again a molecule moves from areas of higher concentration to areas of lower concentration. But facilitated diffusion requires a protein which acts as a channel to allow the protein to move from one side of the plasma membrane to another, and the molecule moves down its concentration gradient. So obviously facilitated diffusion requires a membrane as well as a channel protein, and I think this slide calls it a transporter protein, not a transport protein, but a transporter protein. Let's take a look at that again. Simple diffusion is the movement of a solid molecules from areas of high concentration to areas of low concentration. Like if I were to put a drop of dye in this beaker of water, in this region would be high concentration, and the molecules will move this way to less concentration. As I mentioned, for cells, it's important that simple diffusion explains the transport of oxygen and carbon dioxide into the cell by oxygen and out of the cell by carbon dioxide. Any questions about simple diffusion? Another part about simple diffusion is it can happen with or without a membrane. Like oxygen diffuses into the cell through the cell membrane. But here, this drop of dye is diffusing into the water without a membrane. Any question about any of that? Facilitated diffusion is similar to uh, simple diffusion, but it does always involve a membrane and a transporter protein or channel protein, allowing the molecule to move from areas of high concentration through the membrane to areas of lower concentration. Glucose is not a good example of facilitated diffusion. I'm going to explain why in a little bit. A good example of facilitated diffusion is actually the movement of fructose across the cell membrane. We have a channel protein which allows glucose, like if you eat a bunch of fruit, you have a bunch of fructose in your gut, it'll move into the cell through a channel protein using facilitated diffusion. And then once the concentration of the glucose inside the cell, excuse me, not glucose, fructose equals the concentration of the fructose outside the cell, the net movement of fructose will no longer move into the cell. It won't move out of the cell. There will be no net movement of fructose. Any question about that? So no matter how much fructose you eat, you'll never digest all of it because once it equals inside the cell, you cannot move more fructose into the cell. Any questions about any of that? I am oversimplifying it a little bit because this fructose could move from this cell into the blood, in which case more fructose could move into the gut, into the cell lining the membrane of the gut. Glucose doesn't work this way, except when the glucose is higher outside the cell than inside the cell. Glucose will move into the cell by facilitated diffusion through a channel protein. But once the glucose equals the concentration outside the cell to equals inside the cell, this channel protein changes to an active transport protein. And then that active transport protein will move all the molecules of glucose in the gut inside the cell against the concentration gradient. And this active transport has to have one ATP molecule for each molecule of glucose. It moves against its concentration gradient. So no matter how much glucose you eat, you'll digest all of it. And you can see why the glucose isn't a good example for facilitated diffusion, because when the glucose is higher outside the cell, it does use facilitated diffusion. But once the glucose equals the concentration outside and inside the cell, that facilitated diffusion protein, let's call it a channel protein, changes to an active transport protein, actively transporting the glucose molecules into the cell. In both simple and facilitated diffusion, solutes are transported, and they transport down a concentration gradient from areas of high concentration to areas of low concentration. Osmosis is the third passive process for moving molecules against a membrane or whatever. Osmosis does only happen with the movement of water, where water molecules move down its concentration gradient, moving from areas of high concentration to areas of low concentration. But water is not a solute. Water is a solvent. And osmosis is always when water, the solvent, moves across a membrane. And it always moves passively from areas of high water concentration to areas of low water concentration. Any questions about osmosis? Now the water moves across the cell membrane from areas of high concentration to areas of low concentration. However, the solute concentration will be opposite the water concentration, meaning if the water concentration is high outside the cell and lower inside the cell, the water will move inside the cell. But if the water concentration is high outside the cell, that means the solute concentration is low outside the cell, and the solute concentration inside the cell is high. I'm pointing this out because the tonicity is not defined by the water concentration. The tonicity is defined by the solute concentration. And we talk about the concentration of water by tonicity. A bacterial cell can experience three kinds of osmotic solutions: a cell in isotonic solution, a cell in hypotonic solution, and a cell in hypertonic solution. These are relative terms, so it's correct to state the solution outside the cell was hypertonic relative to the solution inside the cell. Any question about any of that? The easiest one to discuss is isotonic. That is where the concentration of the solute outside the cell is equal to the concentration of the solute inside the cell. So remember, the tonicity is defined by the concentration of the solute, but it's the water that moves into the cell. If the cell is in an isotonic solution, there is no net movement of water either inside or outside the cell. And the cell is happy because there's no net movement of water into the cell or out of the cell. Now, what do I mean by no net movement of water? Does that mean no water molecule ever moves inside the cell? That's a question, yes or no. Is anyone going to answer?
Water can still move around, but it's balanced.
Yes, for each water molecule that moves into the cell, one water molecule moves out of the cell. So there's no net movement of water when the cell is in an isotonic solution. Thank you for the contribution there. A hypotonic solution is when the solute concentration outside the cell is lower than the solute concentration inside the cell. So when the solute concentration is lower, that means the water concentration is higher outside the cell, and then there will be movement of water inside the cell. Now, if the cell has a cell wall, it has to be an intact cell wall, the water will move into the cell until the plasma membrane expands to reach the cell wall. Once the plasma membrane has come up to the cell wall, the plasma membrane cannot expand any further. That will limit water moving into the cell, because if the cell membrane cannot expand and water molecule would try to move into the cell, there will be no position for the water molecule to move into the cell. The cell has taken as much water as it can. There's no more room for the water to move into the cell. And that's how the cell wall can protect the bacteria from the bacteria being in a hypotonic solution. The cell wall will limit the amount of water moving into the cell. Any question about any of that? Now, if the cell does not have a intact cell membrane, or an animal cell which has no cell wall, has no intact cell wall, then the plasma membrane can expand, more water can move into the cell, the plasma membrane will expand further, more water will move into the cell, the plasma membrane will expand until it bursts, and then that will kill the cell. And that we call osmotic lysis, when water moving into the cell kills the cell by bursting the cell membrane. That's actually the way penicillin works. It damages the cell wall, and then the plasma membrane can protrude out of the cell wall. Water can move into the cell. The protrusion will grow. More water will move into the cell. The protrusion will grow even further until it eventually bursts, and then the cell will die. Most bacteria live in a hypotonic solution, and that's why they require the cell wall to survive. And that's like obviously most bacteria causing a human infection. The bacteria are not in an isotonic solution, they're in a hypotonic solution, and that's why penicillin G can work, killing the bacteria. Any question about any of that? All right, a hypertonic solution is when the concentration of solute outside the cell is higher outside the cell than inside the cell. When this happens, the water inside the cell will leave the cell and go into solution. That will shrink the plasma membrane from the cell wall, and water will continue leaving the cell by plasmolysis, which generally leads to cell death. Some cells, like E. coli, are pretty sensitive to plasmolysis, and you put them in a hypertonic solution, they don't survive very long. By at least a day, if not sooner, all the cells would be dead if it's E. coli. That's not true for an endospore, but most other cells, you put them in a hypertonic solution, sooner or later, the cell's going to die. Some cells, like Staphylococcus aureus, can survive for a fairly long time in a hypertonic solution, but eventually, even Staphylococcus aureus will die. So plasmolysis is the shrinkage of the cytoplasm or the cell membrane away from the cell wall, generally leading to cell death. And it's because of plasmolysis, or the hypertonic solution, that molasses can stay at room temperature for years and not go bad. Whenever the bacteria fall into the molasses, the water will leave the bacteria, killing the cell. Or if it's an endospore, it just won't ever germinate. And the molasses can stay well at room temperature for years. My mother had a bottle of molasses that we figured was at least 20 years old, and I made molasses candy out of it, and my father enjoyed it. Didn't kill him or anything. Okay? So he ate that molasses that was 20 years old or older. And the bottle of molasses was about 30 years old, we figure, before my mother said, you know, this bottle is pretty old. You're not using it, and I'm not using it, so I'm going to throw it away. So the molasses was probably good at 30 years old. And that's what we estimated. It could have been older. Any questions about hypertonic solutions? All right. So passive processes are okay when the concentration of substance required by the cell is higher outside the cell and inside the cell. But what about a cell in an environment with a low nutrient concentration, like a cell in the ocean? Very few nutrient molecules around the cell. Or a cell in the Sahara Desert. What can the cell use to move the molecules that it needs inside the cell? Well, it can use active transport. So the cells accumulate nutrients against their concentration gradient by using active transport, pumping the molecule against its concentration gradient. This requires energy. The cell needs to perform work to move the molecule against its concentration gradient. It also requires an active transport protein and then one ATP molecule, ATP being adenosine triphosphate, and you use one ATP molecule to move one molecule inside the cell against its concentration gradient. The transport proteins are generally molecule specific, meaning we have a different active transport protein for sucrose than we do for glucose, than we do for lactose. So we have 3 different active transport proteins for pumping in those 3 different types of sugars. Any question about any of that? So obviously we have 20 amino acids in humans. We must have 20 different active transport proteins for the amino acids to bring the amino acid into the cell. All right. Prokaryotes can move molecules into the cell by active transport, and eukaryote cells can only bring molecules into the cell against the concentration gradient by an active transport. However, prokaryotes have another way of moving molecule against their concentration gradient. They can move molecules into the cell by an active transport protein, but they can also use group translocation to move the molecule into the cell against its concentration gradient. Group translocation is a special form of active transport only found in prokaryotes. It does require a transport protein. It does require a high energy phosphate compound, but not ATP. It's something else you don't need to know. PEP's an example. And then it requires a molecule to move into the cell against its concentration gradient, such as glucose. But that glucose molecule, once it is transported into the cell, is chemically altered. The phosphate, which comes off of PEP, which is giving the energy that the transport protein needs to move glucose against its concentration gradient, the phosphate comes off of PEP and then is chemically altered the glucose. So we have glucose 6-phosphate enter the cell on the inside, and then glucose outside the cell. So glucose will be transported into the cell, but once inside the cell, it's chemically altered to another molecule. In this case, it's glucose 6-phosphate. You don't need to know that, but I'm just giving you an example. All right, let me show you why. What would be a good one to do? Probably paint. Where it is. So here's our cell. And then we have low concentration outside the cell of a molecule. See if I can write this. Oop, kind of messed it up, but there we go. Then we have an active transport protein, so let's put that here. And let's say we have, let's go to this now. Actually, let me erase that here, because I need the room. Let's say we only move the molecule in by active transport. And let's say we have 10 molecules inside the cell. Let's say 10 molecules of glucose. And we have, I guess we should make that 20 molecules. I'm going to erase that. I might not be able to. Well, kind of see it. Let me erase both of them. Twenty molecules inside the cell and 10 molecules outside the cell. We're talking about glucose. I'm just going to go G for that. All right. So when you move the first one in by active transport, you're moving it um, one molecule against 20, which what would that be? 10 against 20, something like that, which is against the concentration gradient. But when we move the last one in, it's now 1 against 29, meaning the concentration gradient is steeply increased. With group translocation, let me move that up a little. Let's say we move 5 by active transport. So, oop, not working. So the last one will be 1 versus 25, which is bad, but it's not as bad as 1 versus 29. And then the other 5 are moved by group translocation. And here we're going to be 1 versus 4. And it's going to be a glucose 6-phosphate inside the cell. This is a much less steep gradient than 1 to 29, 1 versus 4. You see why the cell, the prokaryote, uses group translocation? Because it makes it easier to move the molecule into the cell, and then also easier to retain the molecule in the cell. And then 5 of the molecules move by active transport, and then 5 move by group translocation. Any questions about that? Well, hopefully you got that. And that allows the cells to accumulate substances easier when they use both group translocation and active transport. All right. The cytoplasm is the substance inside a prokaryote cell. In a eukaryote, remember we have cytoplasm and nucleoplasm, but in a prokaryote, all there is inside the cell is cytoplasm. Cytoplasm is mostly water, about 80%. It does have dissolved solutes in it, like carbohydrates, glucose, even DNA will be dissolved in the cytoplasm. Where the DNA is called the nucleoid region, but there's no boundary between the nucleoid region and the cytoplasm. This The nucleoid region is wherever the DNA is floating in the cytoplasm. There can also be other molecules inside the cytoplasm which are large enough that they are not in solution, like a ribosome is large enough it does not go into solution. And then inclusions, these are molecules the cell is accumulating in the cell, and they sometimes become so accumulated that the molecules come out of solution, precipitate, and then we see a solid mass in the cytoplasm, and that is called an inclusion. And there we have an inclusion there, and there's one there, an actual transmission electron microscopic image. Any question about any of that? All right, remember the nucleoid region. There's no membrane in between the nucleoid region, the cytoplasm. It's wherever the DNA is floating in the cytoplasm. So the nuclear region is a part of the cytoplasm. It just tends to stain differently. You can also have plasmids in the cytoplasm. A plasmid is a short molecule of DNA. The chromosome is a very long molecule of DNA. The chromosome has thousands, maybe even hundreds of thousands of genes on it. The plasmid only has a handful of genes on it. So it's a much smaller molecule. You can have ribosomes in the cytoplasm. Remember, a ribosome is large enough that it does not go into solution. You can actually see the dots there, those are ribosomes. The ribosomes are the protein factories, meaning that they make proteins in the cell. You don't need to know the makeup of a ribosome, but you should know that prokaryote ribosomes are a little smaller than eukaryote ribosomes. Prokaryote ribosomes are 70S in size. Eukaryote ribosomes are larger. They are 80S in size. You don't need to know too much about the S. The S is actually the sedimentation coefficient, where the ribosome sediments in an ultracentrifuge gradient. And that's how biochemists and molecular biologists measure the size of ribosomes. They centrifuge it in an ultracentrifuge gradient. And that's what the S stands for. Ribosomes are made up of ribosomal protein and ribosomal RNA, also called rRNA. Inclusions, remember, they are reserve deposits within the cytoplasm. Material the cell is accumulating. When the material becomes so concentrated that it begins precipitating out of solution, then we call it an inclusion. So you have a solid mass of material the cell is accumulating. Because there's so many molecules, it can no longer be suspended in solution, and so it precipitates out. You don't need to know that there are different names and different types of inclusions, but this slide is showing you that. Like I said, you don't need to know it. All you need to know is that there are different types of inclusions. Here are some pictures of various inclusions, magnetosomes, sulfur granules. The cell is accumulating sulfur, probably because it metabolizes sulfur. And then this yellow line is the cell, and there are metachromatic granules in this cell, 3 of them, and that one and that one. All right, another thing you find inside the cell, at least in some prokaryote cells, are endospores. Endospores are a specialized resting cell produced by certain species of bacterium, mainly species of Bacillus and Clostridium, those two families. The endospores are highly resistant to desiccation, heat, chemicals, radiation. An endospore can survive conditions where the vegetative cell cannot. So the vegetative cell will be the cell that made the endospore. You also call it a parental cell. The parental cell is metabolically active, growing and reproducing. The endospore is not growing, is not reproducing, and is essentially metabolically inactive. And that's why it can be highly resistant to desiccation, heat, chemicals, radiation, and survive conditions the vegetative cell cannot. Sporulation is sporogenesis, the endospore formation. Like these cells are making endospores. There's an endospore inside a parental cell right there. And then germination is the return to the vegetative state, meaning this endospore, when the environment becomes favorable, the endospore can germinate and grow into the vegetative state, a cell having metabolism, growing and reproducing. Any question about any of that? We'll talk about endospores in 3 places in this class. I don't remember if this is the first time we've talked about endospores or the second, but we will talk about them a 3rd time. This is one of the topics that's so important. We'll talk about it at least 3 times in the term, so you can count on a question about endospores on both one of the quizzes and also the final. Any questions about any of that? All right. An endospore happens when the cell goes through cell division, but unlike normal cell division for most bacteria, the cell does not divide evenly down the middle. It divides into a smaller portion and into a larger portion. So first the DNA will divide, and then one molecule DNA will move towards this side of the cell, the other towards this side of the cell, and then the cell will start dividing, making a smaller cell and a larger cell. The smaller cell will form a plasma membrane around the DNA, and the larger cell will really just have the cell membrane form. This one, we don't call it a daughter cell, we call it the continuation of the parent cell. It's immaterial what you call it, but this is the parent cell making an endospore, and they still say that's the parent cell making the endospore. The endospore then moves inward a little bit, and it gets a second cell membrane put around it. So endospores have 2 cell membranes put around it. And then a peptidoglycan layer forms between the 2 cell membranes. The spore coat is then put on the endospore, and now the endospore can survive harsh environmental conditions, and the parent cell will eventually die. When the parent cell dies, it releases the endospore. Any question about any of that? That's endospore formation. I have a video here, but it's not very good. Let me see if I can get it to play. Well, that's nice. No volume there, but that's all right. Let's skip it. Oh, come on. There's no volume here, but there's the endospore forming. I guess that's not really that good. And then that's it. All right, that's the end of the prokaryote cell structures. No, it isn't. There's a nuclear area. I thought I talked about that. The nuclear area is the nucleoid region of a prokaryote cell where the DNA is located. It's wherever the DNA is floating in the cytoplasm. I already talked about ribosomes. Maybe I talked about all of these. Yep. All right. So this slide is showing you the cell structures of a plant cell, shown on the left here, and then the cell structures of an animal cell, shown on the right. You should realize that some structures found in one are found in the other, like a flagella can be found on a plant cell. What's an example of that? There's a fern. I think it's a firm sperm, but I'm not sure what you call it. A firm pollen maybe, that has a flagella. Obviously, a sperm cell is a cell on humans that have a flagella. If anyone can name another human cell that has a flagella, go ahead and name it. I think the trachea. No, that's cilia, I think. Wait a minute. Is the trachea flagella or cilia? Don't remember. Sorry, I used to know. I just don't remember. Anyways, sperm cell is a human cell which has a flagella. Let me blow this slide up so I can see it better. Yep, that's now out of. Yep, well, that's good enough. Um, guess I'll stay there and just blow it up this way. Chloroplast is an organelle that is only found in plant cells. You don't find chloroplast in animal cells. The centrosome is something you find in animal cells. You do not find it in plant cells. Oh, excuse me here. Hopefully that'll stop me from sneezing a second time. I tend to sneeze twice. Uh, the mitochondria is something found in both plant cells and animal cells, the nucleus in both plant cells and animal cells. The nucleus is the largest organelle in an animal cell, but the largest organelle in a plant cell is actually called the central vacuole. You can find a vacuole in a plant cell, but you can find vacuoles in an animal cell also. The vacuole is much smaller than the central vacuole. Anything else I need to point out? A vacuole is a transport organelle, transport vesicle. It's a transport vesicle that is moving something from one area of the cell to another. And if the transport vesicle stops moving and then starts storing what's inside it, that we call a vacuole. The central vacuole is a very, very large organelle, the largest organelle in a plant cell, that is both a storage area for molecules the cell wants, as well as a storage area for molecules the cell does not want, like storing glucose that the cell is making can be put in the central vacuole, and then the uric acid. They don't have urea because it's a plant cell. I think it's uric acid, which is an nitrogenous waste that the cell has. Cells don't pee, so they store those waste in the central vacuole. And the central vacuole is kind of odd because imagine you're storing your food and things you're going to eat in the refrigerator, and then you store your waste from the toilet as well in the refrigerator. That's just the way plants are. They store a whole bunch of things in the central vacuole, both waste material and then things the plant cell actually wants. Any question about any of that? All right. Comparing a prokaryote cell with a eukaryote cell, I'll let you go through that. Eukaryote cells can move by one of three means. They can move by a flagella, they can move by cilia, and then they can also move by pseudopods, like a white blood cell moves by pseudopods. Let me show you that eukaryote cells can have cytoplasmic streaming. Where the movement in the cell actually moves the organelle in the cell, or the molecules in the cell obviously move. Come on. Cytoplasmic streaming. In this case, it's the chloroplasts which are moving, but that's just because they're visible in the cell. You can see they're moving around the cell. Here they're moving this way. Any questions about cytoplasmic streaming? I think that's it. Remember, an organelle is a membrane-bound organelle. In eukaryote cells, you only have membrane-bound organelles in eukaryote cells. And this is showing you, well, these are membrane-bound organelles and non-membrane-bound organelles. I call non-membrane-bound organelles cell structures. I want to talk a little bit about the mitochondria and the chloroplast. These are special organelles because they have two membranes. They actually have DNA inside them, and they have a DNA molecule inside these organelles, and they have ribosomes inside these organelles as well. We'll talk about why in just a little bit. Eukaryotes have an endomembrane system where molecules can be made in a ER, endoplasmic reticulum, and it can be a lipid in the smooth ER or a protein in the rough ER, but the molecule will be made, and then it will be packaged into a vesicle, a transport vesicle, which will generally move to the Golgi apparatus for modification. And then the molecule will be modified in the Golgi apparatus and then packaged and sent out the Golgi apparatus on the other side of it in a transport vesicle, where it will be transported to where the molecule needs to be. If the transport vesicle just moves somewhere and then stops moving and then starts storing the molecules inside of it, we call that a vacuole. But the transport vesicle could move to the cell membrane and then fuse with it and then dump the contents of the transport vesicle outside the cell. And those of you who are experienced teenagers, you'll be seeing teenagers make hormones, which they'll transport outside the cell and then dump it outside the cell into the blood. And you can see these teenagers with raging hormones. Any question about any of that? Anyways, that's the transmembrane system which the molecule is made in the ER, either the smooth ER for a lipid or the rough ER for a protein. It will be packaged into a transport vesicle, fused into the Golgi apparatus, be modified in the Golgi apparatus, and then come out the other side of the Golgi apparatus as a transport vesicle, and then go to where the cell wants to have that molecule. And this example shows the molecule going outside the cell, but it could be inside the cell, or could even be just stored in the transport vesicle inside the cell. In which case it's not called the transport vesicle because it's not transporting, it's now storing, and that's why it's called a vacuole. Any questions about any of that? All right, so let's talk about the endosymbiotic theory, the theory to explain the evolution of mitochondria and chloroplasts in eukaryotes. The theory is, a long time ago, we had a proto-eukaryote, meaning a cell that was before a eukaryote and it later became a eukaryote. And we had a bacteria which was capable of aerobic respiration. So let's go over here. Make it a little bigger. That means this bacteria could take one glucose and make 38 ATP. The proto-eukaryote, on the other hand, only could make ATP by fermentation. Make it a little bigger that way. There we go. That means one glucose could make two ATP. Oop. Well, a long time ago, the proto-eukaryote engulfed the bacteria. I'm just going to make pseudopods on this. So that one, I guess this one. So it surrounded the bacteria with pseudopods, and these pseudopods grew and grew. Get the eraser here. Till the pseudopods eventually surrounded the bacteria, meaning the membranes here fused. And then the bacteria was brought inside the cell, which it actually is inside the cell now. You'll note two things about this bacteria. First of all, it has its own cell membrane, and then the membrane of the organelle bringing the bacteria inside the cell. So this has two membranes now, a cell membrane and or an organelle membrane around it. This cell has DNA in it, the original DNA, as well as ribosomes. This bacteria took a glucose, which, why isn't that showing here? I don't know why I'm losing the left of my thing there. All right, let's see if we can move that over. I probably can't. Oh, are you gonna have to do proto-eukaryote? It's supposed to be 2 glucose. Maybe I can erase that and put in another 2. Glucose from the proto-eukaryote diffused into the organelle membrane, diffused into the cell membrane, and then was taken up by the bacteria. The bacteria took that glucose and made 38 ATP molecules. And then for some reason, the bacteria share the ATP with the proto-eukaryote. Probably life was early and the cell had lots of glucose. The cell might have had lots of ATP. And so they just allowed the molecules to diffuse out of the proto-eukaryote and into the bacteria, and then the ATP out of the bacteria into the proto-eukaryote. We don't know how much ATP the bacteria gave to the proto-eukaryote, but it was a lot more than 2. And then the proto-eukaryote was a bit confused and said, that should be 1. Glucose makes 2 ATP. Sorry, let me erase that again. You should correct me when I make a mistake like that. Oop, don't want that. I think I'm going to just erase this whole thing. Oh, that's good enough. Kind of read it. 1 glucose, 2 ATP. The point is that the bacteria gave it a lot more than 2 ATP. The proto-eukaryote was confused. It lost 1 glucose, but gained a whole bunch of ATP. A lot more than the prokaryote can make from 1 glucose. So the proto-eukaryote kept the bacteria around to make it ATP. And the two started living in symbiosis with each other. They've now lived in symbiosis for so long, we do not find this original bacteria outside the proto-eukaryote. And the proto-eukaryote changed. It now became a eukaryote containing a mitochondria, which now makes ATP for the eukaryote cell. And that's the theory about how the mitochondria evolved. In plants, this happened twice. Actually, it happened 3 times. We'll talk about the third if I have time today. But it did it with the mitochondria first, and then it engulfed a photosynthetic bacteria, which became the chloroplast in the plant cell. So that is our theory for how the eukaryotes gain the mitochondria and gain the chloroplast from literally initially engulfing a bacteria, probably to eventually eat it, but it kept the bacteria around for a while, and the bacteria made it ATP. So the eukaryote cell kept that bacteria around to make it ATP because the bacteria can make much more ATP than the proto-eukaryote could. So what evidence do we have to support this theory? First, we have some eukaryotes, which are protists, which are simple eukaryotes, would actually engulf a bacteria. And there's some amoeba species of protists which lack a mitochondria. And what these amoeba do is they engulf a bacteria and then have that bacteria make the ATP for the amoeba. So the amoeba lack a mitochondria, and they go out and find a bacteria, which it takes specific bacteria, probably one species, and then will engulf it, and then they start living in symbiosis. The amoeba feeds the bacteria glucose and other nutrients that the bacteria wants, gives the bacteria a home, and then moves around. And the amoeba makes, not the amoeba, the bacteria makes ATP for the amoeba. So that's our first one. Here we're looking at a protist, and it's engulfing an algae, and then the algae will make glucose from sunlight and carbon dioxide, as well as other nutrients the protist gives it, and share some of the sugar with the protist host. And then the two live in symbiosis. So we actually see that happening today. Some other evidence is that the mitochondria and the chloroplast have two membranes, an inner membrane and an outer membrane. And we predicted that if they were an engulfed bacteria, where do I have that? That they would have two membranes. Where's my paint? Here it is. It'll have the organelle membrane when the proto-eukaryote brought the bacteria in, and it'll have the cell membrane. And in the mitochondria, we have the outer membrane and the inner membrane. The inner membrane being the one making the ATP. Some other proof is the DNA of a prokaryote is usually one circular molecule. In bacteria, they lack histones. They're circular molecules. The eukaryotes have linear molecules like the X chromosome. They're not circular. They have multiple chromosomes. The mitochondria and the chloroplast have one circular chromosome. And this chromosome lacks histones. The prokaryote or the bacteria lack histones, and the eukaryotes have histones on their DNA. The first amino acid made in a eukaryote cell when we're making a protein is methionine. In a prokaryote cell or bacteria, the first amino acid is formylmethionine, a different amino acid, but a similar one. And then the first amino acid made in a protein in the mitochondria and the chloroplast is formylmethionine, the first amino acid in the protein. The ribosomes of a eukaryote cell are 80S in size. Of a bacteria, they're 70S in size. Of a eukaryote, mitochondria or chloroplast, the ribosomes are 70S in size. You look at the growth, eukaryote cells divide by mitosis, prokaryote cells mostly by binary fission. The mitochondria and the chloroplast can divide by mitochondria and chloroplast. And then the strongest evidence is if we sequence the DNA in the mitochondria and the chloroplast, they do not fall in the domain eukaryote. They fall in the domain bacteria. Any question about any of that? All right, that's the endosymbiotic theory, the theory about how cells obtain mitochondria and chloroplast. And I want to briefly state that they're actually watching another organelle evolving in plant cells. It is a nitrosome, a bacteria that can fix nitrogen, has been living in symbiosis with a plant cell, and it's on the way to becoming a organelle inside the plant cell. Any questions about any of that? All right. If there's no questions, Start the lab at 6:31, giving you 10 minutes. Bye.